Mitosis. Interphase, consisting of 3 stages, G1, S, G2. During G1, the cell grows and prepares for synthesis. During S phase, synthesis, or the copying of DNA, occurs. And lastly, in G2, it prepares for cell division. The stage G2 is being shown in our diagram. Here, the chromosomes cannot be seen individually and is in the form of chromatin. The centrosomes have formed and the nucleolus is visible in the chromatin. Prophase. The chromatin begins to condense into chromosomes, and the chromosome consists of sister chromatids, which are attached at the centromere. The nucleoli disappear and the nuclear membrane breaks down. Also, the centrosomes move to the polar ends of the cell, and microtubules and spindles begin to form. Metaphase. The centrosomes are now at the opposite poles of the cell, and the sister chromatids have aligned in the middle of the cell on the metaphase plate. Anaphase. The sister chromatids have broken apart and each has its own centromere. Each one now forms a chromosome. So now, the 2 opposite chromosomes move toward opposite ends of the cell by the microtubules, and the cell has twice the number of chromosomes. Telophase. 2 daughter nuclei form, and the chromosomes have now reached opposite ends of the cell. The nuclear membrane and the envelope reform, and the nucleoli reappear. The spindle fibers have now gone away, and the chromosomes have become less condensed. Finally, during cytokinesis in an animal cell, the cell forms a cleavage furrow which helps pinch the cell into 2 cells. The division of the cytoplasm takes place and forms 2 genetically identical daughter cells. The importance of mitosis is that mitosis and cytokinesis produce the somatic cells that make up our body. The process of producing these cells is a continuous cycle to generate new cells and replace dead and damaged ones. During mitosis, 2 genetically identical cells are created and 46 chromosomes are produced in each cell nucleus.
Meiosis 1. Prophase 1. Chromosomes begin to condense and form loose pairs. Tetrads form and the arms of the sister chromatids begin to overlap and are held together in synapsis, which allows for genetic diversity. Each pair has chiasmata, where crossing over has occurred. Lastly, spindles form and the centrosomes move towards the poles. Metaphase 1. The pairs of homologous chromosomes are now aligned on the middle of the cell on the metaphase plate. Both chromatids of one homolog are attached to the kinetochore microtubules from one pole. Anaphase 1. The homologous chromosomes separate and move toward opposite poles. The sister chromatids are still connected at the centromere, which causes the chromatids to move as one unit towards the same pole. Telophase 1 and cytokinesis. The 2 haploid cells form and each chromosome still consists of 2 sister chromatids. The cytoplasm divides and forms 2 separate haploid daughter cells, with each cell consisting of 23 chromosomes. In animals, a cleavage furrow forms to help the cytoplasm divide. Meiosis 2. Prophase 2. The spindle fibers form and each chromosome consists of 2 chromatids, which are attached at the centromere. Metaphase 2. The chromosomes align in the center of the cell along the metaphase plate. The kinetochores of sister chromatids are attached to the microtubules that extend from opposite poles. Since crossing over occurred during meiosis 1, these 2 sister chromatids are not genetically identical. Anaphase 2. The chromatids separate and move toward opposite poles as individual chromosomes, each consisting of their own centromere. Telophase 2 and cytokinesis. The nuclei form and the chromosomes decondense. The cytoplasm begins to divide. 4 daughter cells are produced with a haploid set of chromosomes. Each cell is genetically distinct with 23 chromosomes. The importance of meiosis is that the resulting 4 haploid daughter cells, which are genetically distinct, contain half the genetic material of their parent cells. These cells are known as gametes, which are used for sexual reproduction. Meiosis allows there to be heritable variation among the offspring. Meiosis ultimately results in 4 daughter cells, and each has 23 chromosomes.
Meiosis 1 versus Meiosis 2. Meiosis 1 involves the separation of homologous chromosomes, while Meiosis 2 separates sister chromatids. Also, Meiosis 1 results in 2 haploid cells, and Meiosis 2 results in 4 haploid daughter cells that are genetically unique. Also, more specifically, during prophase 1, crossing over occurs where the homologs have formed tetrads, and overlap occurs at the chiasmata.
Mitosis versus meiosis. First, mitosis is the process of producing new somatic cells that are used to replace dead and damaged cells. Mitosis is also a process of growth and development and part of asexual reproduction. The mitotic cycle produces 2 diploid genetically identical cells that have 46 chromosomes each. Conversely, meiosis is the process of producing gametes, which are sex cells that are used for sexual reproduction. Meiosis produces 4 haploid cells that are all genetically unique due to crossing over of DNA, and each cell consists of 23 chromosomes.